Very good morning to all the viewers who have joined us from all over the world. Good morning, good afternoon, good evening, depending on where you are at this time. We have an excellent speaker, Professor Fanus Venter, who works at the Department of Biochemistry, Genetics and Microbiology at University of Pretoria. He's been associated with the Burgess Manual Trust for a long time and has been a part of the BISMiS since the day it was formed. The title of his talk today is The Irrelevance of Prokaryotic Subspecies. Fanus has been very active in the field of bacterial evolution, systematics, and diversity. He's specifically interested in knowing how these aspects of bacteriology can be applied to answer specific research questions related to the evolution and functioning of bacteria and bacterial communities in different environments. During the past few years, his research group has focused on two main types of areas. The first one is bacteria associated with plants, that's rhizobia, and the second one is aquatic environments, especially drinking water and salterns that he is focused on. He also serves as an associate editor of the IJSEM, as well as the Systematic and Applied Microbiology. He is also the South African ambassador of the International Society for Microbial Ecology, that's the ISMI, a council member of the South African Society for Systematic Biology, member of the International Committee of Systematics of Prokaryotes, and a secretary of the executive board of the Seed Code Committee. With this brief introduction, I can assure you that the session today on the irrelevance of prokaryotic subspecies will be very enlightening and will open up a new Pandora for discussion that is to come ahead. So Fanus, the screen is all yours.
Okay. Yeah, as Kamlesh, thank you very much for the invitation to speak at BISMiS. It's all a BISMiS related event. It's always a great pleasure for me to be involved. As I've earlier said in our discussion, that BISMiS meetings has always been the highlight conferences that I've attended, because you have people that are interested in the same things, and you can have quite lively discussions. This is also the place where ideas are born and where we evolve those ideas. And today I'm going to talk about an idea that has also evolved through the years, and specifically connected to BISMiS meetings and things. So yes, as you've said, it's quite a Pandora box to say prokaryotic subspecies are irrelevant, but I want to sort of take you through the way that I thought about it and the reasons why we feel that it might be the case, and that we need to look at this. So I'm at the University of Pretoria, as you've said, and have also been quite involved with the BISMiS itself. So to take us back, so as you've said in the introduction, my interest is really in evolutionary biology and how that relates also to systematics. So I also have always have an interest in how would evolution explain things. And it started with my trying to understand what is species and how did evolution form species, and you will hear some of that in it. From there, it progressed into what is subspecies, and then in 2018 we had the BISMiS meeting in South Africa, and there you can see Kamlesh as well. We had some African activities like drumming and dancing and things like that. But at that conference, I presented a talk, which was the start of this journey that I had on what exactly are bacterial subspecies. So that was the question that I asked at that time. And from there, then we had further discussions, me and my co-workers, and we came up with this publication in 2022, where we said, well, the relevance of prokaryotic subspecies in the age of genomics. And I need to stress clearly that this work was done together with a co-worker of mine, Emma Steenkamp, and at that time, one of my PhD students, Marika Palmer. She's now gone to the States and will move to Canada, where she will, I think, be at the University of Manitoba soon. So we had quite long discussions on this and developed the idea. So if you want to follow what what I'm going to present today, you can also follow it by reading the article. It's probably better described there than what I will do today, because in producing an article, you have the words and you can really clearly make your statements, but it's open access, so you can have a look in new microbe and new infections for this specific publication. But once we went through it and made our statements and followed the argument, we actually came to the conclusion that subspecies should be considered to be irrelevant. And I want to take you through that today, why we are of that opinion. But first, let's have a look what exactly are subspecies. And this is a slide still from that BISMiS meeting. And what we said is, if you grew up in Africa and you used to blue wildebeest, you might be able to see the differences in the subspecies of blue wildebeest occurring in sub-Saharan Africa. But if you look at them in a different way, from just a few color changes and morphological things, you would not be able to distinguish them. So the one thing that we need to realize is when we look at subspecies, it's always with a specific lens. We have this specific characteristic that we find. And in the eukaryotic world, of course, the biological species concept and the idea of mating and gene exchange is quite important. Now, at this stage, you can see there are maybe some differences between these animals. But if we look at bacterial subspecies, it feels to me that I'm not sure what to look at. Where are the variants that you find which is still linked to within that species pool? So, but let's start at looking at subspecies. As I say, zoology was the zoologists were the first group of people to really start looking at subspecies. It was already in 1789, they said that varieties could be seen as subspecies, and it really referred to phenotypic variations within a species, often linked to geography, because in zoology, you might find that populations are separated from each other and you don't have interbreeding happening. So certain traits become more pronounced within a certain group. But the moment you bring them together, you will find that there can still be crossbreeding between these populations. Charles Darwin also referred to subspecies, and he says it represented incipient species and its variation in isolated population, which is not part of a continuum. So those are the ideas that are quite commonly used in zoology. The one thing is, if we think part of the debate on subspecies, this debate is much more contentious within the realm of zoology, botanists as well, but zoologists really, there's quite a lot of papers on discussing whether we should have subspecies and what is the benefit of subspecies. Now, the latest in that literature, especially linked to the zoologist, is a paper by Kevin de Quiros, and he's quite known for his idea that there's a single species concept which you could use, And then he made he described his point of view in this article. And what he stressed is that subspecies in zoological terms is really when you have incompletely separated lineages. So some interlineage exchange can still happen. And so what it means is basically that there's not a reproductive isolation, and therefore, if you apply the biological species concept, this will not, it will be considered to be one species, but because of small differences, you can consider them to be parts of that and therefore a subspecies. But one of the statements that he made in that paper is that subspecies are actually species. There is no difference between a subspecies and a species. The only thing is that there's still a limited amount of gene exchange between them, so they're not totally isolated populations, and therefore, but as I say, he clearly made the statement that subspecies should be species. Now, if we now move to our group of species of interest, we sit with a bit of a different scenario. We have species, but we know that, the core genome is only the things that are shared by every member of the species. And you have the pangenome, where you have a number of accessory genomes which are related to either niche adaptation or specific phenotypic differences. And the major difference in our case compared to zoologists is that horizontal gene transfer is actually rampant, especially linked to the pangenome. The only reason the pangenome can exist really is because of horizontal gene transfer. And so we as taxonomists have to deal with this issue in a bit of a different way from what we find in for the zoologists or the botanists, for instance. So how we've gone in bacterial taxonomy, talking about archaea and bacteria, the species definition or description depends typically on 3 traits. So it says that there should be phenotypic cohesion, based on measurable and observable traits. It should be a monophyletic lineage, and genomic cohesion. So based on a nucleotide similarity measurements or so, you would say that these genomes are quite closely related. But the one thing that we need to look at is the issue of monophyly, because that for me is where our argument comes in, and you will see that in a moment. So this is how we typically go about describing species at the moment. Now, let's go and have a bit of a look at what has been said about subspecies in terms of bacteriology. So the first variations that's been noted and linked to subspecies has been metabolic variations, and it was by Rogosa, specifically variation in the lactic acid bacteria. But please note that this was a phenotype, a measurable, observable phenotype that could be used to distinguish them. Again, medical and technological importance was used to make these describe these variations or subspecies. So we have different medical conditions that could be associated with these, or strains that could be used in biotechnology, etc. The one thing that we need to also take note of is that in the original code, they spoke about varieties, that you could use varieties, and if these things are very stable and clear, you could call them subspecies. But then subsequently, Rule 5C made it clear that varieties are exactly the same as subspecies. So it actually took, although you could use varieties, they say, well, these things are actually subspecies. So that's what's currently in the code. That's how the original use of these things were. Then with hybridization and sequence data coming along, we know that it's changed the views. And one of the important ones is the ad hoc committee, the Wayne paper in 1987. And in there, they said there's some evidence that subspecies is a concept that is phylogenetically valid. And this, I've highlighted it in red because this is really where the crux comes in. They say, well, subspecies are linked to something that you see in terms of the evolution of these organisms. It's not just variation, specifically, phenotype that can change, but there is also an underlying evolutionary basis. And then in a Brenner article, they say the designation of subspecies is at the pleasure of the investigator. So again, saying that, some people will use it and some will not. So what is the current approach if we look at how people define subspecies? So typically it's based on either digital hybridization or MLSA data or ANI data. So that's usually the basis. And then in the Chun article on the use of genomics in taxonomy, they also said, discrete genomic centers of variance, coherent and distinguishable clades, and delineation is based on the fact that these organisms share a higher than 95% ANI or a digital hybridization of more than 70%. So, and they also said that there should be a sound rationale, for instance, host specificity, or one of those traits that you could use to support subspecies. But the one thing which is clear here, and this is how it's often used, is people use the 95% or the 70%, depending on which technique they use, as a cutoff. So when you see things that are sort of different and fall, but if the 95%, then they use it as a very clear cutoff. And we've seen articles recently where people have described species and a set of strains as different species, and then people got genomes, they did it, they said, oh, it's above 95%, and they suddenly make these subspecies instead of keeping them as a species. So that's the trend that we see at the moment, and that's one of the things that I feel we should address. If we look at the number of subspecies that's been described for the last 20 years, and this is data based on the LPSN, they've got some very nice data sets that one can use. You can see that when we had MLSA, and that was over the whole 20 years, there were constantly people saying these groups are different and they can be separated. But the moment we started having genomes, we had quite an increase in the number of subspecies. For instance, we had 55 in this specific year, which is the most that we had in a year. So we can see that people are quite actively using this. And I think the main reason for this use is that 95% cutoff that are seen as a very solid barrier. You need to be either on the one species or subspecies based on that specific characteristic. Now, if we compare on how we compare species and how people compare subspecies, we see that we use similar criteria in terms of phenotypic cohesion. We say they should, a subspecies should also have shared phenotype. Species should have it. They should be monophyletic. And as I said in that Wayne paper already, they spoke about, there's a lineage that might distinguish them. So, which immediately tells us that there should be a notable genetic isolation of the core genome, that the core genes which we use for our species delineation should be monophyletic. They should already have split, and there should be limited gene exchange in that. The only criteria, as I've indicated here, would be the 95, whether it's 95 or not. And that is where I start having a problem, because as I said previously, we started looking at, well, how should we define species and what is really a bacterial species. And our approach is an evolutionary approach. So we say we need to look at how they have evolved. And in a paper published in 2019 and some work that we've presented at the previous BISMiS conference, we have said that all bacterial species are uniquely evolving groups. They sui generis. So they're not all the same. This is a continuous process. So they didn't all start the same. They have the evolutionary age of species might differ, and the population sizes might differ. So you might, in a small population, you get that certain traits get quite quickly enhanced, whereas in a widely distributed one, there's a bigger gene pool that are shared and more sort of equalization between these. And our main point was that the boundary between species and populations should be determined individually for each species. So we say, look at the data, and if you see that they are the core genome are quite well separated, you always have a little bit of gene flow between these. But if the common message is that these things have evolved separately for quite a while already, you should call them a species, whether they make the 95% or not. And we have in the past described, motivated that and described some species where we had values higher than 95, because we could strongly show that this is the case. So if you want to have more information on how we consider and look at species, you can go back and listen to a BISMiS talk that was given by Marika Palmer in 2021 on species concepts, because that clearly explains the way that we think and how we feel species should be considered, and also bringing in the whole issue of genealogical concordance, where you look at individual genes, and that helps you to determine that border between what is a species and what's within species. Just as an example, I want to show Klebsiella quasi-pneumonia as an example, and in it, this is already a paper about 10 years old. They've described 2 subspecies. And when they did this description, if you look at the paper, you will see that these 2 subspecies are phylogenetically distinguishable clades, which is already something that we find typically for species as well. There are stable phenotype differences between them, but they have an ANI value of 96.4%. And therefore, they were not considered to be 2 different species, but as subspecies. So the moment people start looking and describing subspecies, you already get the idea that, you know, that they've met all the hallmarks of a species. It's just this one thing that they want. So subspecies are often well-defined entities, and we should keep that in mind, because we believe that we should, why call them a subspecies? They've separately evolved. They might just be younger, and therefore we can call them a species. So if you look at the MLSA for these 2 subspecies based on the number of isolates, you can see that there's a clear distinction between the 2 groups. And in the paper that we did on the relevance in new that I've showed up front. We've described some of these examples in Klebsiella, where some of the subspecies should actually, in our mind, be raised to species, and in other cases there are subspecies that are just variants in terms of phenotype, and therefore should be remain as the same species and not be called subspecies. Okay, so this is what we want to look at. We want to look at the evolution, see that there's a clear distinction based on core genome. You cannot do this based on the accessory genome, because many of these strains might miss out on a specific gene. So we always look at genes that are commonly shared between these organisms. But if you also do ANI, and depending on how you want to sort of group them, you can see that they do make a very clear, distinct clusters, that they share amongst each other much more higher sequence identity than between these two groups. And therefore, we are of the opinion that this should be considered to be species. But then let's get back to this the idea of the pangenome, and how do we then deal with the pangenome? You know, we say, okay, for core we use species, but we know that there are quite a lot of interesting variations within species that we want to describe. It might either be things, as we said before, that are niche adapted, or it could be phenotype that are shared among certain groups. And the one thing we should realize is that many of these traits are of quite interest to the wider microbiological group of microbiologists. For instance, a trait like nitrogen fixation in the rhizobia, nodulation, those are things that sits often on the accessory genome, but are quite important. There might be pathogenicity factors that are variable, and we want to be able to also deal with that. We don't want to use a subspecies for that, because we believe that these things are not phylogenetically supported, but we need to find a way to use it. And what we're saying is that looking at the accessory genome, it underpins the divergent phenotypic characteristics, and we can enrich and enhance taxonomic descriptions by using the basis of varieties or biovarieties, such as biovar, pathovar, serovar, symbiovar. So this is something that's been in use in many cases, and we feel that we should strongly support that to describe the within species variation linked to specifically the pangenome. So just to make the clear distinction, what we say, if we have evolutionary distinct groups with specific characteristics, we should describe them as a species because they are a lineage on their own. If we have a species within a species variation, we should use meaningful characteristics for the end user. This is very specifically, the end user wants to be able to differentiate between these, and therefore we should use meaningful descriptions as described in the code in Appendix 10. One of the things, and I'll just talk about it a little bit later as well, is that in some of these variations might overlap. So you might find, if you look at a biovar, a certain strain would belong to a specific biovar and another one to a different biovar. But if you look at pathovar, you might find that both of them belong to the same pathovar. And that's fine, because it's to use this variation in a specific context that we want. It's not a fixed taxonomic description. It's really a useful way of dealing with these variations. So if we look at some of the examples that we've included in that paper, you have pathovar, for instance, in E. coli, Pseudomonas syringae, symbiovars for the nodulation in the Bradyrhizobium, Mesiorhizobium. You have serovars, well-known serovars, like, for instance, in Salmonella, and then morphovars and biovars, where you have very specific biological characteristics. So these categories are already known and could easily be used for the description within the variation within species. So what, in our mind, are the benefits of having varieties? The first thing, it really focuses on observable variation, and it highlights the importance of the accessory traits. So many of these, as we said, many of these observable variations are linked to the accessory, and therefore you would describe it as varieties. They convey very important biological information. So we say we should really use this, but it should be based on the need of the end user. It's already established practice, as we've shown. People have used varieties quite a lot in certain fields, and we feel that we should, it's established practice that we can just enhance. And the one thing also, it's transferable across species. And this means that, symbiovars could be shared between different species. For instance, they have the same nod genes to be able to interact with the legumes, but they belong to two species, but it means that you can call this a symbiovar, and two species could have individuals belonging to that same symbiovar. So it's really catering for a broader variety, not only mainly within species, but you could also have it for between species. And then what would be the potential impact of demolishing or stopping to use subspecies? The first thing, we realize that it will have an impact. And the first thing would be that all subspecies will have to be reassessed. We need to have a look, is this not really a species? And they should be then changed in their description to species. But there are also some that's just a variation, a phenotypic variation. We know in Klebsiella pneumonia, there are actually subspecies that are not easily distinguishable based on the genetic information, and therefore it's really a accessory genome thing. And in those cases, you would rather go for variation than specifically calling them subspecies. It would mean that many of the databases would have to be updated. We know that in the GTTB, they are not using any subspecies delineations there. They just go up to the species level, which is fine, but they use a 95% cutoff or for grouping of these things. And therefore, if you have a species that are higher, you would need to interact with the database and say, well, please consider this. We've got enough proof to consider this and deal with it. Of course, if we want to abolish subspecies, we will have to adapt both the bacterial code and the C code, because both of them allows for the description of subspecies. We believe that having no subspecies would give us a better reflection of evolution and speciation, because we will really be able to highlight that some of these things, although they might share a lot, have evolved separately, and we can start looking into what are the drivers, what were the selective criteria that make them go apart. And the one thing, especially implementing the use of varieties, is we will make taxonomy more accessible to the end user, so they can really start using this. So then, finally, I just want to sort of summarize what we had, and this is also a figure from our paper, what we say is, if you look at the core and you find that there are monophyletic groups that are characterized by high levels of phenotypic and genomic coherence, call them species. The whole issue of a genus is another Pandora's box, something that I feel this is probably the next thing that I need to address in my mind. I have clarity in my mind at the moment of what to call a species. I don't want to call anything a subspecies, but I'm not sure what I need, how exactly I feel about genera. Well, I have some ideas, but I think we need to start having this conversation, a similar conversation as the one we're having today on, what defines a genus. Then, if you have accessory related characteristics, we want to call that varieties, and it can be host association, biochemical traits, or typically growth traits, etc., biovars, etc. So this is what we're proposing, and as I say, the main message is, if we look at how our idea of subspecies have evolved over time, it has actually we have a convergence with species, and therefore we're actually calling the same thing by two different names. And my feeling is it's irrelevant. We should just use one name. And with that, I thank you.
Thank you, Fanus. That was really enlightening and creates a new thought process in the mind. Actually, if I'll continue with my question first, because I think I need to have a clarity for all the audience that are listening to us. So, to my mind, when we use the word species, whether it is in the term subspecies or a species, it's an evolutionary term. And probably that's the reason whenever somebody wants to define a subspecies or a species, it must have an evolutionary context to it. And probably that's one of the reasons, because when you say that accessory genome derived content in an organism is most likely because of the adaptation in the environment. And that's why it's not an evolutionary concept, it's more of an adaptation, and it's a temporary phase. It can be lost when the organism is taken out of the context. So that's why you want to propose that instead of calling them subspecies, it's a variety.
Yeah.
Right in my understanding?
Yes. Yes. So what we say is those things that are variable, but that are linked to the same core. So that's the critical thing is, if you look at the core, it's two different groups. But if you look at the accessory, it might be shared between them, or only part of that, the ones having that same core would have that characteristic. And therefore, it's a variation within a species, or a variation that we're interested in terms of a wider group of organisms, but it's not a fixed thing and really linked to the core genome.
Right. Yeah. So I think that also allows a certain bit of clarity for our listeners, because some of the I asked this question because some of the questions that have come up in the Q&A box actually are still not clear about whether it's an evolutionary context or the adaptation context. So I'll start with the questions that have come from the listeners. The first one is on similar lines. It's from an anonymous attendee, and the question is, any strain as isolated may differ in brackets, more than 5% when the same strain is domesticated. So it's like when a same strain is isolated, when it's a wild type, it becomes a unique one. But when it is domesticated in the lab, it loses some, it gains some, and that's why it changes in the content. So how you encounter or, collectively answer this kind of an issue?
I'm a bit confused about the more than 5%. I'm not sure 5% of what, because typically what we've seen is, yes, it loses things, but if you do, for instance, an ANI, it will still be quite high. It might, it will still be 99%, not 100%. So, for a 95% difference, it's a major difference. And that's not something that I think can happen in the time span of you having something in the lab. The 95 differences we've seen have often come over sometimes million years. If you date these species, you see that they have a estimated history of sometimes some million years, and it might be even 40 or more million years. So I'm unsure on 5% of what. It might be 5% on a specific gene, but if you look at the whole organism and the genome, you might find that it's I think it's not that 5% difference that we see.
Right. Aaron makes a comment about
the 55. Yes.
Yeah.
I've just went to their data and looked at the number. So that's what I could see. Unfortunately, I cannot vouch for why that has happened. I can look into that because it was also quite surprising to me. I think there might have been some of the larger papers that's been, especially I think the one on the alpha proteobacteria, where they've suggested things. But I'm not, unfortunately, I took that data as, and that's why I've said this is the data that I got from the tables in the LPSN. I was also quite surprised, but I didn't have time to go into individual ones and find out why that was the case.
Because the audience is not able to see the questions, I'll just repeat about the numbers. Aaron has a comment that he's confused about the number of subspecies described in recent years. The number 55, which is added in 2020 as based on LPSN information. And as per his information, which he checked on LPSN, he just found 10 new subspecies, which were added in 2020, whereas 1,166 new species were added in the same year.
Yeah. So as I say, if you go into LPSN, they've got a section where they show graphs. And based on those graphs, so they showed you a bar graph, and you could actually see, new validated species, and they indicated, what the different groups was. So that, we'll have to look at that, Aaron.
Yeah. The next question from Aaron is, as a journal editor, I regularly see manuscripts based on one colony, one new species kind, where authors had spent time and effort to characterize a colony and then concluded that they had not a good case to describe a new species. So they decided to call it a subspecies instead, so that they still could publish a paper. This is surely not what the concept of subspecies is for.
Yeah, well, I fully agree. I think in terms of if you really want to describe a species and you end up with singletons that are sharing more than 95% similarity, which we know that the below 95 typically belongs to different species. I think that is something that we're not contesting. We've seen that often. If you are below, then things are already well separated. It is these more recently evolving species that are higher, and if you then end up with a value, I would just not describe it. I would fully agree with Aaron, especially if that ANI value does not support it. And for our concept of species, we actually want more isolates, because then we can look at how they perform in individual genes and where that cutoff is of gene sharing and no gene sharing. So it's an important thing.
Right. Ian has a question about genera. What do you think of the rarely used category of subgenus?
I think anything above species is actually should be useful.
Mhm.
It has, in my mind, no real evolutionary basis. Yes, we know that they've split and they've evolved, etc. But I think any category. My personal feeling is I can find a species and differentiate it from a different one based on certain characteristics. But I find it difficult to know how to differentiate between genera, and subgenus would be something that I have. I think I don't like it. Why would you have another arbitrary defined level that you want to bring in? Sometimes, if you have a subgenus, why just not call it a genus? Another genus that's closely related. That would be my take on this. But yeah.
Right. An anonymous. Yeah.
Sorry, just to come back to the whole issue of genus. I think for those higher taxa, we really need to think, what is the impact on the end user? Because we are also dealing with taxonomy that should be used by other people, and we should make it accessible to them. And therefore, splitting genera and things might, when it's an arbitrary thing, when you split these things based on certain characteristics and it's not useful to the end user, it doesn't make sense. We've seen that with the mycobacteria, for instance. There was quite a backlash because from the user community on the splitting of some of these genera.
Right. So there's an anonymous attendee who's asked a question. Please correct me if I'm wrong. The nomenclature of variety is not regulated by ICNP, but the nomenclature of subspecies is regulated by ICNP. Then why not just keep using subspecies instead of variety? Aaron has already, made a clarity on this question. He says that see Appendix 10 of the ICNP for information about infra-subspecies specific divisions that are not regulated by the rules of the ICNP.
Yeah, so it's not regulated, and I think it should not be regulated because this is a user-defined thing that you want to use. It's not a recognizable thing that you want to have a very specific name to it. That's community driven, user community, end user driven, whereas species we want to sort of have, this is very different species specifically. Right.
The next question is also, it's not in the context of the talks today, but Jay Ganesh wants to know whether the candidatus phytoplasma and spiroplasma are under C code or ICNP or any specific code.
Well, the candidatus will be under the ICNP, but only with if it's a candidatus, it only has an effective name. So it has not validly a name that has been validated. At the moment, under the C code, all the names that have been validated has been brought into the C code, but candidatus has not yet been looked at. But it might be, if there is a good genome for it, that people might be able to describe it under the C code and get a valid name for it. But that's right. a different conversation.
Another question coming up from an anonymous attendee is, if my research does not look at the rank of subspecies, then the names at the subspecies rank can be left aside, right? I can just report the species name, especially when I don't need to distinguish the subspecies. Likewise, if I'm studying specifically at a species, having clades that have specific subspecies names would be useful, isn't it?
Yeah, I think you need to look at your specific group of organisms and then have a look at the subspecies and why they've been called subspecies. If it's some of the older subspecies like we have in Klebsiella pneumonia, it might be based on phenotype that doesn't gel with the evolutionary history that we see. But in other cases, a subspecies might already actually be a species. And if you want to do that comparison, you should include them because they actually represent a very closely related one to the group of organisms that you're looking at.
Right. There's just some last two comments by Ian. Regarding numbers, Ian says you could try a Web of Science search with subspecies.nov.
Yeah.
Keyword. That can be done. And Ian thanks Fanus for the subgenus may have some use when splitting genera is controversial, as in the case of Mycobacterium.
You could, as I say, you could use it if it keeps the user happy and you want to distinguish that there are lineages in it. So, but it becomes to a way of describing variation within a genus or lineages within a genus. So, in my mind, it's what they want to call it that drives this. And if people are happy with it, you can always indicate that there are lineages, but why give it a very specific name that's valid in the code? Then just use the genus and say you have these specific lineages within this genus, and they might have different characteristics. If they are really phenotypically different, I think it makes sense to call it a different genus. That
right.
I also want to say is, there should be good basis for doing this. And if you feel that it will help the end user by making this differentiation, then we should actually do it and people will start using it because there's value to it. And that's the importance in terms of genus, I think.
Yeah, I think for everything, the usability for the end user, sort of has become a point of concern, especially whenever taxonomic changes happen in the nomenclature of an organism. That is bound to create a confusion in the end user's mind. And
yeah, we always get people never interested in taxonomy until there's a change, and then suddenly they voice their opinion, which is fine, but we would have loved them to be involved all the time. So that, it's important. I think it's also important that we as taxonomists also work within the field of the things that we describe so that we have an understanding of what the implications of this would be. And sometimes we're lucky as microbiologists because we don't get funding for taxonomy. We get funding for these applications, and then we can do the taxonomy and we're already embedded in that field, which makes life a bit easier compared to some of the other, zoologists or so that are just basically taxonomists and nothing else. But I think we need to always keep the end user in mind because we want it to be a system that's useful because that's the only reason we created this.
Right. Yeah. Thank you, Fanus. We had a full discussion, and I'm sure the viewers will go back to presentation when it goes live tomorrow, and they will have more questions and they'll get connected with you.
Yes, people are welcome to contact me. If you go to that paper, you also find my email address there, but it's quite easy to get hold of me.
Yes. Thank you, Fanus. And to all the viewers, we'll see you once again on the third Saturday in the month of August. And hope to see you all register for the BISMiS 2023 and become a participate in the elections. Thank you.
Yes, BISMiS meetings are very nice. You should attend.
Thank you.